Can you wiggle your ears?
Jiggle your eyes.
Do your fingers bend in strange ways?
And what about your arms?
Can you make your knuckles dance?
Not everyone can do these things. Whether or not you can is all determined by your genes. To find out how, let's learn more about genes, genetics, and DNA. Each of us is a unique individual. From the color of our hair and eyes to the shape of our lips. How is it that no two of us is exactly alike? That's because each of us is a combination of traits passed down to us from our parents. How does that happen? To find out, we have to go back billions of years to the first life forms to appear on Earth.
Those look like bacteria.
They are. And the first life forms were similar to modern-day bacteria. Tiny microscopic organisms, each consisting of a single cell.
Aren't all living things made up of cells?
Right. And bacteria are the smallest, simplest cells. Yet as simple as they are able to perform one remarkable feat. Can you guess what that is?
Multiply.
That's right. They are able to make exact copies of themselves, one after another, after another.
How do they know how to do that?
To find out, let's split this model of a bacterium in two. The inside of a bacterium is filled with a jelly-like fluid. Located within this fluid is a thin, ribbon-like loop called a chromosome, shown here in red. The chromosome carries information controlling all the cell's activities, including reproduction. There are hundreds of separate bits of information on the chromosome. They are referred to as genes.
So when a bacterium reproduces, it is actually following hundreds of separate instructions, or genes.
That's right.
They all look identical.
They are. That's because they have only one parent. The same is true for other single-celled life forms, like these microscopic pond organisms. They have only one parent. The same genes are passed down generation after generation. That's why all of these organisms are identical. But look at these kittens.
Aww, they're cute.
They're from the same litter, yet they're all different. They have different colors and different markings. That's because they have two parents and share the genes of both. Single-celled organisms need only one parent to reproduce. But most multicellular organisms, that is, organisms with many cells, need two parents in order to reproduce. Multicellular organisms include just about every plant and animal you're familiar with, including insects. You've probably seen insects connected at their abdomens.
Sure.
This is how many insects mate. In this example, the male mantis inserts a special organ into the female and injects a fluid into her abdomen. The fluid contains specialized cells called sperm. The female has specialized cells of her own. They're called eggs. When an egg cell is joined by a sperm cell, it will develop into a new being in a process called fertilization. These are human sperm cells. They're extremely small and can be seen only under a powerful microscope. The cell's nucleus is found in the head of each sperm.
Nucleus? That's like the cell's control center, right?
Right. Since sperm cells are so small, we'll use these models to show them better. All of the male's genes are contained in each cell.
Where are they?
They're found here in the head, on its chromosomes, inside the cell's nucleus. There are 23 chromosomes in all, and all the male's genes are contained on those chromosomes. Human egg cells also have 23 chromosomes. Males produce many sperm cells, and each seems to race to reach an egg. But only one will be permitted to penetrate an egg. When a sperm cell is successful, the 23 chromosomes of the male sperm cell join the 23 chromosomes of the female egg cell, creating a new cell with 23 pairs of chromosomes.
For a total of 46 chromosomes.
The fertilized egg cell is called a zygote.
So how does a single zygote become an entire organism?
By reproducing itself, in a process called cell division. This type of cell division is called mitosis. You end up with two identical cells.
Each with 46 identical chromosomes.
Then these cells will reproduce themselves, and so on and so on. These are just models of cells. This is a real cell. It belongs to a salamander, and it's about to undergo mitosis. Watch what happens to this thin line here. It's a membrane that surrounds the nucleus.
It disappeared.
It's one of the first stages of mitosis.
What are those things that look like worms?
Those are the chromosomes. They've recently duplicated, and they're moving to the center of the cell.
They look like they're lining up.
That's exactly what they're doing. They're getting ready for one of the most important parts of mitosis. Now watch closely, as the chromosomes seem to pull apart, almost like taffy, and move to opposite ends of the cell.
Neat.
Finally, the cells separate.
And you end up with two identical cells.
What about the dimples? You guys have dimples?
Your height, the shape of your nose, your complexion, everything that makes you a unique individual are all passed down to you from your parents. These are all referred to as traits.
But what determines which traits will be passed down?
That was the same question asked by Austrian monk Gregor Mendel more than 100 years ago, and he decided to find the answer. Working with pea plants, Mendel learned several important rules about heredity. One was that an individual carried two factors, or genes, for each trait, but passed down only one. Another rule was that one of the factors, or genes, was dominant over the other.
Dominant?
That means it has more influence or power over the other. Let's use a simple example to explore these rules. If this black rabbit and white rabbit were to mate, what color do you suppose their offspring would be?
I don't know. Black?
Maybe white?
How about gray?
No need to guess. To find out, we'll use a simple diagram called a Punnett square. We'll assume there'll be 4 offspring, 1 for each square. 1, 2, 3, and 4. Next, we'll put the black rabbit here on the left, and the white rabbit on top. According to the rules discovered by Mendel, we know that each rabbit carries 2 genes that control color. Let's say the black one is purebred for black.
Purebred?
That means both of its genes are black. We'll refer to them as capital F, capital F. F is for fur. The white rabbit is also purebred. That means that both of its genes are white. We'll refer to them as small f, small f.
Which genes will the offspring inherit?
Since the black rabbit carries only 2 black genes, capital F, it can only pass down a black gene to each one of the 4 offspring. Since the white rabbit carries only 2 white genes, small f, it can only pass down a white gene. Each of the 4 offspring end up with both a black gene and a white gene. But each will be black because black is dominant. They may look exactly like the black parent, but there is a big difference. These offspring are no longer purebred, but are now called hybrids. They carry both a white gene and a black gene. The black trait is dominant, and the white trait is recessive.
Recessive?
That means it's hidden.
Well, what would happen if 2 hybrid rabbits were to mate?
Let's do another diagram using a Punnett square. This offspring will receive a black gene from the parent on the left and a black gene from the parent on top. It will end up capital F, and will be purebred black. This offspring will get a black gene from the parent on the left and a white gene from the parent on top. It will end up capital F, small f, but will be black because black is dominant. This offspring will end up with a white gene from the parent on the left and a black gene from the parent on top. It will end up small f, capital F, but will be black because, again, black is dominant. Finally, this offspring will end up with a white gene from the parent on the left and a white gene from the parent on top. What color will it be?
It has only white genes, so it has to be white.
Good. Now, which offspring gets which gene is completely up to chance. All we know for sure is that about 1 in 4 will be white. None of the rabbits will be gray, by the way, because in this example, these traits are not blended. The same rules of inheritance apply to many of the traits found in all plants and animals. If you were to cross hybrid purple corn with hybrid yellow corn, what would be the result?
That would depend on which color is dominant.
Right. And since purple is dominant, the ears would have kernels that are about 75% purple and 25% yellow. 3 to 1. So far, we've only talked about a single trait, color. But all plants and animals inherit thousands of separate traits from their parents. Thousands of separate instructions that help determine what and who we will become, from the way we look to our talents and natural abilities.
Talent is inherited?
If you're good in sports, music, or art, chances are you inherited that talent from your parents. These 3 girls are triplets, and each is an accomplished musician, just like their mother. But even though you inherit talent, you still have to learn how to use it. And that's why hard work, dedication, and training are so important. Mrs. Julie Hogarth is a teacher at Miller South School for the Visual and Performing Arts in Akron, Ohio.
Many children inherit certain characteristics and talents, but part of what Miller South is here to do is to teach the children how to use those talents. And one of the ways we teach them is to fine-tune and teach them the skills to use their talents. When you first start learning to play the piano, you learn the notes. When you first start learning to paint, you learn about color. Those are skills that are fundamental that we build from. Although oftentimes talent is passed down, skills aren't passed down and must be developed. And that's one of the things we work on here at Miller South.
So much is determined by our genes, even whether you can wiggle your ears, jiggle your eyes, or bend your arms in strange ways. All of which are a result of inheriting 2 recessive traits, by the way.
It gets stuck like this sometimes.
How is it possible for so many instructions to fit inside every human cell?
Oh, good question. For many years, chromosomes were something of a mystery. We knew that they were made of deoxyribonucleic acid.
What was that?
Everyone calls it DNA for short.
That I can pronounce.
We knew that DNA carried our genes from one generation to another. What we didn't know was what DNA looked like or how it worked. That wouldn't come until the 1950s, when scientists Rosalind Franklin, Francis Crick, James Watson, and Maurice Wilkins found the answers that unlocked the secrets of DNA. Finally, we were able to look deep inside the cell, into the nucleus, into the chromosomes themselves, to see exactly how DNA carried the blueprint of life from generation to generation. This is a model of a DNA molecule.
Kind of looks like a ladder.
It does, but this ladder is twisted. The rungs of the ladder are formed of 4 chemical bases that may be arranged in a wide variety of ways. We may refer to the 4 bases as A for adenine, T for thymine, G for guanine, and C for cytosine. You will only find A joined with T and G joined with C. It is all in the way these chemical bases are arranged that determine the characteristics of a life form.
4 bases don't seem like very many.
No, but remember that a DNA molecule has millions of rungs, so there are nearly endless combinations possible. And it's the way they're arranged that determines every one of our traits and make us unique individuals. In fact, they determine whether or not organisms become amoebas, or zebras, lions, or dandelions. DNA also holds the secret to how chromosomes are able to make exact copies of themselves in a process called replication that occurs just prior to cell division. It is during replication that the DNA molecule splits down the center of the ladder. As it splits, additional bases are called in from the cell nucleus. The bases attach themselves to the sides of the split ladder in the same way they were in the original molecule. The result will be 2 identical DNA molecules. With this knowledge, scientists are now able to do what was thought impossible just a short time ago. They're now able to remove a gene from a DNA molecule and replace it with another in a process called genetic engineering. Genetically engineered bacteria are being used to make drugs to treat disease. Farmers are now using genetically engineered bacteria to help fertilize their fields. Genetically engineered crops like cotton and corn grow bigger and better. Genetically engineered salmon grow 3 times faster than normal ones. With more animals and crops like these, we could wipe out starvation throughout the world. Genes and DNA molecules are difficult to see, but scientists are able to identify them by using special images. These so-called DNA fingerprints show the genes that line a DNA molecule.
How many genes do we have?
As a result of the Human Genome Project, we learned that each of us has more than 30,000 genes. This knowledge has led to major breakthroughs in medicine, because just about every disease we know of is influenced by abnormalities in a person's genetic makeup. At labs like this one, tests are being done on human tissue to help doctors and their patients make important medical decisions, even about conditions that may not become apparent for months and years. Here, human cells are being examined to determine whether there are any problems in their chromosomes or DNA. Powerful microscopes are used to visually inspect the chromosomes themselves.
In this test, we're looking at abnormal chromosomes in order to find something that might be missing, something that might be added, anything that will tell us what's wrong with our patient. For example, sometimes chromosomes switch material so that one part of one chromosome is on the other part of the chromosome, and that could be a big problem.
The lab also analyzes DNA for abnormalities. In order to do this, the DNA must be removed from cells, in this case from blood cells.
When we analyze your DNA, we take a blood sample and blow apart the white blood cells. We purify the DNA from that, and we might use a technique called sequencing, which breaks down your DNA into the very specific base pairs or chemicals that make up the DNA ladder. After that, we'll compare that to a normal sequence and look for the little mutations or mistakes that may be in there that might explain why someone has a condition or might be at risk for a condition.
By finding out about medical problems early on, doctors and their patients are better equipped to make important decisions, including treatment considerations. So far, labs like these are only able to identify abnormal genes. In just a few years, they hope to be able to replace bad genes with good ones.
What our lab does is to diagnose genetic disorders. The hope is that in the future, that for the single gene defects that are diagnosed, that gene therapy will be able to be used to cure them.
Gene therapy is a type of genetic engineering and involves the removal of a problem gene and replacing it with a healthy one. This therapy may save millions of lives in the future.
Genetic engineering really sounds great.
It can be, but we should be careful. Some people are worried that the medicines we create to fight disease might have a serious side effect of which we're not yet aware. And others wonder whether genetically engineered food is completely safe to eat. And others think it's just not a good idea to change a person's genes.
Should we be concerned?
Most scientists think the benefits of genetic engineering outweigh the risks. So, what have we learned?
We've seen that the traits of organisms are passed down from generation to generation through genes.
And that the genes are found on the chromosomes within every cell.
When single-celled organisms like bacteria and protozoa reproduce, they make exact copies of themselves because they have only one parent.
When multicellular organisms reproduce, they hardly ever make exact copies because usually the genes of both parents are shared by the offspring.
Genes are passed down during the process of fertilization, when a male sperm cell containing the male's chromosomes penetrates a female egg cell containing the female's chromosomes.
Human egg cells have 23 chromosomes.
Human sperm cells also have 23 chromosomes.
During fertilization, the 23 chromosomes of the male sperm cell join the 23 chromosomes of the female egg cell, creating a new cell called a zygote with 23 pairs of chromosomes, 46 in all.
This new cell will then reproduce itself during a process called cell division, or mitosis, creating two identical cells, each with 46 chromosomes. Then these cells will reproduce themselves, and so on and so on.
Many traits are passed down according to rules first discovered by Gregor Mendel, who learned that they are passed down in pairs and that one is dominant over the other.
It wasn't until the 1950s that scientists discovered what DNA looked like and how it was able to reproduce exact copies of itself through a process called replication.
Scientists have mapped more than 30,000 genes carried by humans.
Changing the genes of organisms is called genetic engineering, and it has started us on an exciting journey.
Where that journey will end, no one really knows.
That's for sure.